Hey guys, in this video we are going to revise the biochemistry from first aid. So let's start with the molecular biochemistry. So in molecular biochemistry, first the structure of chromatin. We know that in our cell there is a nucleus, and nucleus contains this nucleic acid, that is DNA, in all eukaryotes, and the main function is to pass the genetic information to another generation. So first see the DNA. So DNA, it is a condensed and in chromatin form. We know it is very long, so obviously it has to be in condensed form to be packed into the cell, and it is in chromatin form. And how actually it is packed into the cell. So let's see. So first the DNA is going to loop twice around a histone. So there is histone protein and DNA. Histone has basically four types: H1, H2, H3 and H4. So this H2, H3 and H4, this three is going to take part in the formation of nucleosome. So in the center there will be this histone protein, and DNA is going to loop twice around the histone, and this will form a nucleosome. And the remaining histone type, that is H1, it will bind to the nucleosome and to linker DNA, thereby stabilizing the chromatin fiber. So DNA will loop around twice around a histone octamer to form nucleosome, that is beads on a string. Why it is called octamer? We know that it is a histone. So this four kind of protein, H2A, H2B, H3 and H4, they are twice, so they will form octamer and form this nucleosome, and it is known as beads on string. Plus H1 binds to the nucleosome and to linker DNA, thereby stabilizing this chromatin fiber. Now DNA will be negatively charged and histone will be positively charged. DNA will be negatively charged due to phosphate and histone. Histone are large, so from large you can remember lysine and arginine. So lysine and arginine will give them positive charge. Now during the mitosis, it will condense and form the chromosome. We will come to this part, but first let's see the two kinds of chromatin. We have learned that it is a chromatin form. Now it has mainly two types: heterochromatin and euchromatin. So heterochromatin, as you can see here, it is slightly more condensed than this euchromatin. So heterochromatin is condensed and euchromatin is less condensed. It is condensed, so obviously it will appear dark on electron microscope, and it is condensed due to increase in methylation and decrease in acetylation, and that's why it is transcriptionally inactive. And euchromatin is transcriptionally active. Now we have to learn about this methylation and acetylation. So we have to learn DNA methylation and histone methylation, acetylation and deacetylation, and how it is going to affect the transcription. We will again then come back to this part. Okay, first let's see 4 points. So DNA methylation, it is reversibly changes the expression of DNA segment without changing the sequence. So we don't have to change any sequence. It is going to remain as it is. We are just going to do the methylation, and it will reversibly change the expression. As you can see here, reversibly changes the expression of DNA segment without changing its sequence. DNA methylated imprinting. So methylation within gene promoter typically repress, silence the gene transcription. So you can remember that methylation makes DNA mute. So that's why it is involved in X chromosome inactivation, then carcinogenesis, aging, epigenetic changes, genomic imprinting, and transposable element repression. So this was about DNA methylation. Basically remember that it is a reversible process. It is going to make the DNA mute, so transcription won't happen. And when exactly do we need this? So in X chromosome inactivation, in genomic imprinting, and certain pathology like carcinogenesis and aging, plus it is a epigenetic change. Now histone methylation, it is also reversible condition usually, and transcriptionally suppresses, but can also cause activation depending upon the location. So histone methylation also mostly going to suppress the transcription. So you can remember DNA and histone methylation together. Usually causes reversible transcription suppression, but can also cause activation depending upon the location of methyl groups. Histone methylation mostly makes DNA mute. And we have learned this lysine and arginine, they are going to cause a positive charge on histone, and they are usually gonna be methylated. So this lysine and arginine will come everywhere if you are talking about histone. So lysine and arginine, here also you have to remember. And yes, DNA methylation dysregulated is implicated in fragile X syndrome. You can also remember this one. Now, histone acetylation and histone deacetylation. So in histone acetylation, there will be removal of histone positive charge. That's why DNA coiling will be relaxed, and opposite is going to happen in histone deacetylation. So they both are opposite process, because acetylation and deacetylation, acetylation is going to increase the transcription and deacetylation is going to decrease the transcription. So this removal of histone positive charge, and histone positive charge is usually helping the DNA has negative charge, so they both are going to bind together, but this reversal of positive charge will cause reversal of coiling and increase transcription. Now histone deacetylation, removal of acetyl groups, that's why decrease in transcription. So this histone methylation, DNA methylation, we have remembered these two together, that it is going to make the DNA mute, and in histone methylation mostly mute. And this is also opposite process, histone acetylation and histone deacetylation. Now about the nucleotides. So nucleotides, we know that it is of two types. And here also you can remember the examples, that in Huntington disease, this histone deacetylation may be responsible for altered gene expression, and the thyroid hormone synthesis is altered by acetylation. So the fragile X, this Huntington disease, and thyroid hormone, these three example you can remember here. Now the nucleotide. So nucleotide are made up of base, then deoxyribose, sugar, and phosphate. And if we are going to remove this phosphate part, it is going to be nucleoside. So base and deoxyribose, sugar, and they are going to linked by 3 days, 5 days, phosphodiesterase bond. As you can see in this, that this is our base, this is sugar, so this is base, this is sugar, and this is phosphate. This is 5' end and this is 3' end. Now first we will learn this base part. So base are mainly of two types, pyrimidine and purine. Purine has 2 rings and pyrimidine has 1 ring. So in purine there will be adenine and guanine. In pyrimidine there will be cytosine, uracil and thiamine. This name you have to remember, A, G and C, U, T. Now how the interconversion will happen? So certain deamination reaction. First let's see about the purine part. So adenine is going to convert into hypoxanthine and guanine is going to convert into xanthine. We know this from the uric acid synthesis, right? Adenine is going to convert into hypoxanthine first and then again xanthine will come, but the guanine is directly going to synthesize into xanthine. So this too is about the purine. This is easy. Now about the pyrimidine's deamination reaction. So first let's see the cytosine. So cytosine is common in DNA and RNA both. So DNA and RNA both will contain the cytosine, but uracil and thiamine will be different. So uracil will be present in the RNA, thiamine will be present in the DNA. So cytosine can convert into uracil by deamination reaction, and 5-methyl cytosine can convert into thiamine. So from this two reaction we can conclude that methylation of uracil will make the thiamine, because here it is 5-methyl cytosine is going to make the thiamine and here cytosine is going to make the thiamine. So this two relation is, if we are going to add the methyl group into uracil, it will form the thiamine. Now the synthesis of pyrimidine and purine part. This is slightly difficult reaction, I guess, de novo pyrimidine and purine synthesis, but let's try to remember this. First let's see the structure and so purine synthesis. What are the 3 amino acid that is required for the purine synthesis? So it is glycine, aspartate and glutamine. So it is aspartate and glutamine, because we know that there is aspargine also and glutamate also. So this can be confusing. So aspartate and glutamine. A with A, you can remember that, aspartate and glutamine. And the first amino acid, that is glycine. Now, so the glycine will give rise to this NC-C bond, and the glutamine will give this 2 nitrogen and aspartate will give N. So from where all other molecule will come? So the folate, tetrahydrofolate, it will give rise to this 2 carbon atom, and 1 carbon atom will come from the CO2. So we are going to number it. Let's see first. So from here, aspartate 2, 3, then 4, this is 5, this is 6. So one ring is complete. Now 7, 8 and 9. So going to write here only that 1 nitrogen is going to come from the aspartate. Second and 8 carbon is going to come from the tetrahydrofolate. Then 3 and 9 nitrogen is going to come from glutamine. 6 carbon is going to come from CO2, and all other, that is 4 fifth carbon and 7 nitrogen is going to come from glycine. So you don't have to remember actually this, just remember this, all other thing is going to come from the glycine. So at aspartate, just from starting from 1A, so 1N aspartate, 2 and 8 tetrahydro. So tetra means 4, so you can remember that 8 divided by 2 is 4, so tetrahydrofolate. Then 3 and 9 is glutamine, 6 carbon, CO2. This was about purine. Now about pyrimidine. So this is the first carbon, first position pin nitrogen, then 2, 3, 4, 5, 6. So it is actually clockwise, naming is. Here it was anti-clockwise. This 2, 3 is from carbamoyl phosphate and all other is from aspartate. So here we have to just remember this only. Now one property we have to remember about this nucleotide, that they are going to absorb light at 260 nanometer and they are acidic in nature. Now about the synthesis part. So purine synthesis has 2 pathways. One is de novo and another is salvage pathway. Salvage pathway and de novo. So